So today's notes will be 10-4. They're on active transport. And if you need a little more help, this is actually from pages 103 to 106 in your book. You'll need to have everything you ever want to know about active transport. So let's talk about it. It is different than passive transport. Obviously, they have different names. For one thing, it requires energy. This one does require the breakdown of stored energy, through the cell's mitochondria to form that ATP, to get those larger or more complex molecules across. So for one thing, these molecules tend to not just easily slide through the biphospholipid layer. But the other thing is they move against the concentration gradient. So this is the opposite of what we've been talking about with osmosis, where everything kind of just evens out and has that homeostasis thing. This is against the grain. This is salmon swimming upstream. This is where molecules move from an area of low concentration into an area of high concentration. They get on to their friends, so to speak. And that's what makes it very different from passive transport. And this is where you get the membrane pumps, these little shapes we've been seeing in all the drawings of the cell membrane. So as you can see here from the inside of the cell, we're not too keen on just sodium ions floating around. We kind of like the potassium ions a little bit better inside the cell versus outside the cell. So here's your cell membrane, and that ATP is used to pump out the sodium and pump in the potassium. So pumps are transport proteins that use energy. These are the main forms of active transport, the main forms of transport that use energy. So protein transfers, this type of protein transfers a molecule from one side to the other. Like I said, this is probably the most common one. It transports sodium and potassium to against, not down gradient. What that means is it's moving sodium, the Na, to where all the other Nas are, and then the potassium, the K, to all of the other Ks are. So there's not a same number of purple circles as on this side as this side, and there's not the same number of silver circles as this side as this side. There's definitely a concentration of one on this side and a concentration of one on this side, which for cells is a good thing. We like it. It's good. So endocytosis is to bring large molecules into the cell. This is kind of like you eating food, for an example. This material is actually engulfed by the cell membrane, and the membrane makes a pouch. So you can see here, this is kind of stage one. It's like, oh, these purple things would probably be better inside the cell. So it makes this little divot, this little pouch, and then it closes off. And the cytosis comes from that cytoplasm kind of sucking it up. And it makes, I don't know if you remember from your organelles unit, but it's a vesicle is what the organelles call. It's a pouch with some kind of fluid or liquid or pigment in it. Plants, their vesicles are filled with carotenoids, ethanocyans, and different kinds of pigments like that. And this vesicle binds with the lysosome. Get a little organelle review. This is actually how white blood cells work. So phagocytosis is a form of endocytosis. So cytosis. Remember that these words are related to each other. And this is in which an immune cell attacks and kills bacteria. So this is your white blood cell, and it actually makes a little pouch around this and it kills the bacteria inside. It almost like eats it and dissolves it, so to speak. Exocytosis is moving large particles outside of the cell. So endo is in, exo is out. Like exo, think exit. Endo, think indoor. So exocytosis, you're taking that vesicle and it's merging with that cell membrane and then opening up to release the gunky stuff out. And that is how you get rid of gunky stuff into the external environment. Anything that you don't want to stay inside the cytoplasm of the cell. Anything you don't want in the cell, to how you release it. All right.